I just want to say to everyone, buenos días, buenas tardes, y buenas noches to everyone who's listening in. I'm really excited to talk to you about this work that we've been working at the LFD and show you some of the FLIM applications. And FLIM, of course, you've been learning throughout the workshop. So this project actually began as a curiosity in terms of trying to understand if we could actually develop a way to look at fluorescence lifetime fingerprints of the developing embryo. And there's a lot of literature that has been published over the past 50 years, excuse me, that has to do with biochemical analysis and looking at the embryo development from the very early stages all the way up through the late developmental stages. And so what is known about the metabolism of embryos during the stages is that they know that from the 2-cell to 8-cell stage embryo, the pyruvate is the major source of nutrient and energy source for respiration. So there's this very nice figure here depicting that the major component is oxidative phosphorylation. But of course, we know that in every single stage, the metabolism can actually switch. And that's what we are looking for, specific markers that told us exactly when the switch would happen. And if we can follow this in real time and real space, we may be able to use the FLIM signature as an index to quantify embryo health. So hexokinase is inhibited during the first stages of the embryo development. And then around the morula stage, there's an increase in glucose uptake. So there's a switch that happens right after the morula stage. If we take a closer look at each of the stages here, and our hypothesis, concluding with the biochemical analysis of the stages of the development of embryo, is that we hypothesize that during the early stages, from the first fertilized oocyte all the way up through probably the 4 to 8 cell stage development, we would see an increase in the amount of NADH that is mostly that could actually dictate the autofluorescence of NADH with the free amount of NADH. So could we look at the ratio between free and bound NADH to see the different stages within the embryo development? NADH is an autofluorescent molecule. I'm going to show you the structure in a second, which is a really nice it's a really nice cofactor molecule because it is actually involved in cellular metabolism. So NADH is nicotinamide adenine dinucleotide. It's fluorescent in the oxidase stage, and then it plays a central role in energy production. And so in the reduced state is fluorescent. In the oxidized form, it is not fluorescent. So that, but we're not looking at NAD+. What has been known in the literature regarding metabolism is that it was actually metabolism can be associated with embryo viability, so the health of embryos. And many studies have shown that embryonic development in multiple mammalian species shows that glucose uptake correlates with embryo health. So that was really nice. It was actually one of the forms where it was hypothesized that if you were able to extract the media that embryos were grown in and quantify the amount of glucose that was left in the media and the metabolites that were produced by these embryos, you can actually quantify whether the health of the embryo was good or not. However, the problem with glucose uptake assays is that they're not very reliable given the number of embryos that are in the petri dish. So all of this has to be carefully quantified and analyzed. So we know that there is a correlation with metabolism and health. And if we talk to a lot of IVF clinics, so this is in vitro fertilization clinics, and we ask them, what is the gold standard for selecting embryos for implantation into the mother? And the idea today, in today's society, where women and partners wait for a very long time before they have children, and sometimes there's an increase in population of infertility across the world, more and more couples are turning into IVF. So this is becoming a very important area in clinical settings. And so they say that the state of the art is just the human eye. So selecting the embryo by looking at the morphology and looking to see the quantity, the number of cells within the embryo, and looking to see if there are any abnormalities in terms of shape of each one of these cells. So that is really it's a good way. The human eye is very selective. However, if the clinician is not trained, or if they're very tired that day, they could pick the wrong embryo to implant, and there's only one chance for implantation. So we thought if we could first follow the metabolism of the developing embryo using fluorescence lifetime imaging, we might be able to have a signature to select the healthy embryo. So nicotinamide adenine dinucleotide is the fluorescent cofactor that we're actually exciting using 780 nanometers in the embryo. And the lifetime of the bound state of NADH is very long. It can range between 1.4 to about 9 nanoseconds. And in the free state, you could see that these two rings here self-quench, and the lifetime is about 0.4 nanoseconds. So the imaging strategy that we developed was to isolate mouse embryos from pregnant moms and put them under a CO2 temperature environmental chamber, and then measure the fluorescence lifetime of each of the embryos. To minimize exposure of the embryo, we decided to take a Z-stack of the entire embryo and collapse the entire Z-stack into one final image format. So about 50 frames of a Z-stack integration took about 3 minutes to complete the image. So we and I will show you some data showing that under these conditions, we did not observe any DNA damage to the developing embryo. So what does the signature of the autofluorescent path look like? And of course, we're only isolating the NADH emission path. And so as you could see here, we're only selecting the emission at 420 to about 500 nanometers. So this is within the blue emission wavelength of light, and we're not looking at FAD. So in this case, what is very unique, first of all, about the signature of the NADH in these totipotent stem cells is the fact that the FLIM signature looks completely different than any other cell that we've ever imaged. As you could see, the lifetime of the signature of all of these embryos here, I'm going to change the lifetime of all of these different embryos are pretty much dominant on this part of the phasor plot. And it took us a long time to understand why the lifetime is very, very different than the lifetime of the metabolic trajectory that has been shown by Chiara Stringari and many other co-workers at the LFD as to the free and bound final lifetime of the signature of the, let's say, the pure species of NADH and the presence or absence of binding. However, what we decided to do, instead of figuring out exactly if we're looking at NADH in a particular bound state, we just decided to look at the differences in the signature for each one of these stages. And in fact, what was really exciting is that each one of these stages actually had a very unique signature. So when we take our cursor and highlight all the pixels that belong to the lifetime here in blue, you can see that the 2-cell stage embryo lights up. And this is the data here. This is the raw data from the 2-cell stage embryo. When we take now our cursor and highlight all the pixels that belong to this lifetime signature, and you could see that the cursor slightly moves over to the right. And so you could see here that that highlights the 8-cell stage embryo. Now there is a switch between the 8-cell to morula stage here, and you can see that the lifetime starts to shift down towards the center of the fluorescence signal, or the phasor plot here. And so as the embryo goes into the late blastocyst stage, it actually, we see that the signature is more or less in the center, where we see all the rest of the NADH lifetimes that's typical in all of the differentiated cells that we've looked at. So endothelial, epithelial cells, and so forth. And so we actually are able to show that using the Phasor-FLIM analysis, we see a movement of the fluorescence lifetime in each one of these developmental stages. And what we wanted to understand is this movement towards the center of the plot. So why is the lifetime of the embryo getting longer? And in fact, you could see that there's a tail here that's going to a very, very long lifetime. And it was also in the literature, if we go back to the literature, we were trying to correlate this lifetime with known biochemical species. And what was known in 2005, there's this paper that was written to show that 8-cell stage embryos have actually a huge amount of reactive oxygen species. And then they decrease in the blastocyst stage. So there's definitely a switch in the 8-cell stage to 16-cell stage to the morula stage, where there's a huge production of the ROS production. And ROS, which is the reactive oxygen species, increases the free radicals that actually are important in development. So this actually has to happen for this embryos to continue development. So there's a very good balance between the production of ROS and maintaining the balance, so it doesn't go too much overbalance, and then the embryo can actually die if it has too much reactive oxygen species. So if we correlate now each one of these signatures with the literature from the 1960s, and just as a reminder, these are biochemical analysis where all of the embryos at each one of these stages needed to be terminated, whereas in our lifetime data, all of these embryos are live. We could see that in the 2-cell stage embryo, the pyruvate uptake dominates. So that means that free NADH should go down. And so when we look at the 4-cell stage, free NADH will go up. So this shift of the shorter lifetime, we believe correlates with the increased free NADH at the 4-cell stage. Now from 8-cell stage all the way up to blastocyst stage, oxidative phosphorylation dominates, and bound NADH levels should go up. In fact, the bound NADH, the fluorescence lifetime of what we get of the NADH signal actually gets much longer, which would make sense in the oxidative phosphorylation where there is an increase of bound NADH in the embryo. So the lifetime actually correlates with the signatures that were done in the biochemical assays, where we see a long lifetime, a shortening of the lifetime, and then it gets long again. So we are very excited to see that our FLIM signature correlated with the biochemical analysis. So we then concluded that the Phasor-FLIM can measure metabolism during embryo development in the implantation embryos. So why is this lifetime getting longer and longer and longer during the reactive oxygen formation during oxidative phosphorylation? We believe that has to do with the fact that reactive oxygen species can actually produce oxidation of lipids. And this was shown by Rupsa Datta, who was a student in Enrico's lab. And what they saw, and this is from their paper, is that the long lifetime species here, which is about 7.8 nanoseconds, correlated with oxidative stress. So the cells here are uptaking oleic acid, and upon the take up of oleic acid, there's an increase of oxidative stress causing lipids to actually oxidize. And we see this very long tail from the signature of NADH. This is, in this case here, this is the trajectory of what we call the metabolic trajectory, which would just mean the difference from free NADH to bound NADH. So it would fall along this line here. However, you see this tail that is predominantly taking over. And if we look at the pixels that are highlighted, this is where the oleic acid is correlating. So we know that the long lifetime species contribution is due to oxidation of lipids. So as we know in the embryos, ROS production is needed for embryo development. In the bovine embryo in the 8-cell stage, the reactive oxygen levels increase and then they decrease by the blastocyst stage. So ROS is really important in this process. So just to correlate the production of ROS, we wanted to get a better, let's say, sense if this is true, what we're seeing with fluorescence lifetime. We can use other fluorescent dyes. DCF-DA is a reactive oxygen species sensor. And so this, the amount of the ROS does not correlate with the intensity of this particular sensor. However, the sensor does tell us if there's positive ROS production here. And so the green is the positive stain at the 2-cell stage compaction, early blastocyst and late blastocyst stage. In fact, we see ROS in each one of these stages. However, the intensity here in the green does not tell us the amount of ROS. So we decided to use the third harmonic generation to look at the amount of lipids that were being produced during the development of the embryo and see if that we can correlate the third harmonic signal with the long lifetime species that we were seeing pixel per pixel. And if we could see that, in fact, the long lifetime species signature in our phasor plot correlated with the lipid using third harmonic, then we could at least infer that the increase of reactive oxygen species was indeed oxidizing the lipids during development. So here we see that at the 2-cell stage, there is no, let's say, localization of the third harmonic lipid species with the fluorescence long lifetime species. However, you start to see the correlation in the compaction, early blastocyst and late blastocyst stage. So we were able to discern that, in fact, this lifetime shift was due to the that amount of reactive oxygen species. So to further look at if we can see at various changes in metabolism, we decided to treat embryos with rotenone and antimyosin A, which are oxidative phosphorylation inhibitors. Now remember that the embryos at this stage are undergoing oxphos, so they have a high bound NADH production, and each one of these is an embryo here. And so my student Ning was able to use rotenone antimyosin A, and we see a shift towards the free NADH lifetime signature indicative of glycolysis. So we indeed were able to manipulate the metabolism to show that the phasor analysis was a very nice way to see these shifts. We can also do the opposite, where we take embryos that are undergoing glycolysis and shift them back to oxphos using 2-deoxyglucose, which is a glycolysis inhibitor. So here now, this was one of the key points that actually allowed us to look at the amount, the lifetimes of the embryos and actually visualize the embryos to see if we could tell if the embryos are healthy and if the signature of the lifetime had changed. So if we look at the 2-cell stage, there's 3 of them here, and they're under 3 different conditions. The first one is a happy media called the casome media, which is highly nutrient additive media. So the lifetime of the casome condition, as you could see, is here. So each one of these points is the embryo that has been incubated on the happy casome media. Flesh holding media is a nutrient lacking media. And you could see the embryo at the 2-cell stage looks fine. However, you can also see that in the lifetime signature, the metabolism has already switched. And under PBS, of course, PBS, we all know it's just phosphate saline solution. And the lifetime, by eye, these embryos look healthy, but already with the lifetime signature, you already see that the lifetime has completely switched from where the healthy embryos are. So this gave us an indication that we can use fluorescent lifetime, and particularly the phasor approach to analyzing this data as a way and a means to identify healthy embryos from unhealthy embryos, even though they looked healthy by eye. So we now developed a pipeline, a strategy for doing this, superovulating embryos, collecting the embryos from the moms, and then imaging them. The imaging, time-lapse imaging, took about 60 hours. And you could see here from the movie, we're able to track each one of these embryos and then fluorescently use FLIM to identify the ones that went up to the healthy blastocyst stage and the ones that arrested in the morula stage. These were considered unhealthy. So we were able to use a way to quantify which one of these embryos was better followed, and making sure that we didn't, you know, the 2-cell stage embryos you could see here. This is the receiver operating characteristic curve to see if we followed 2-cell embryos, they actually, the AUC gave us about 0.73%, which means that the 2-cell embryos were not very sensitive to looking at FLIM. However, the 8-cell stage embryos were very robust, and they gave us very good data analysis at 0.19. Okay, so this is now just finally I'm going to close here with the looking at the distance analysis, and I'm not sure if this was actually given in lectures before. So you've heard about the distance analysis, and I'm not going to go into details, but even though if the FLIM signature by eye looks exactly like they are matching, I can assure you that there's very slight changes to the distribution of the fluorescence lifetime signature here. These are healthy embryos that are viable, and these are signatures from embryos that did not go to the blastocyst stage. So these developed to the blastocyst stage, these did not, but we took the data from the very beginning, so 8-cell stage, and we were able to use this data as this test set to look at the distance to get an index of whether the embryos were healthy or not. So the distance program can distinguish between healthy and unhealthy group at the 8-cell stage. And we have here, for example, a predicted of unhealthy were 17 and predicted as healthy were 9. So this is an easy way for clinicians to see whether the embryo is healthy and whether the embryo is unhealthy. And we're just going to go to the final set here. Our accuracy rate is about 86%. So if you go to the clinic, the grading for looking at embryo health at the 8-cell stage is about 57% accuracy, which means like just the flip of a coin, you could just say yes or no. But with the FLIM signature, we could see that it's about 86%. Our precision is actually really high, it's 92%, and that's the percent of correct classification with respect to healthy embryos. So we were able to image many, many embryos. We had a total of 156 healthy from an unknown set, and 53 unhealthy from an unknown set from our mouse experiments. So now we can add the distance analysis to finalize and have an index for healthy and unhealthy embryo. And finally, we can show that FLIM does not cause any DNA damage to embryos. We know that 2-cell stage are really sensitive to light, so we didn't want to use 2-cell or 4-cell embryos, but you could see that the development to blastocyst still occur at even 3.5 milliwatts. If we use 10 milliwatts of power, the embryos arrested or died. So the morula and blastocyst stage embryos also showed a very good percent in development. And finally, if any of you have any questions, if we're able to implant those embryos into moms, we did implantation studies where we implanted the embryos that were imaged into pregnant moms, pseudopregnant moms. And then we showed that whether we imaged them or not, the embryos gave live births just fine. So we didn't induce any temporary problems to the embryo development. So in summary, the mouse pre-implantation embryo is unique lifetime. We see that the 8-cell stage embryo is optimal for looking at the signature of FLIM. And in our future work, we'd like to investigate the physiology reason for the EBI difference. So in other words, for each one of these stages, what biochemically we're really seeing in terms of the NADH, and look for any chromosome abnormalities such as aneuploidy. So can we identify embryos that have a mismatched number of chromosomes? And then I'll just stop there and thank the people who actually did all this work. I'd like to thank my former student, Ning Ma, who defended her thesis last year, and she's now at Stanford.